back with your next class, and in today's class we are going to start with your second chapter, that is sexual reproduction in flowering plants. So without any delay, let's start the chapter. Now, sexual reproduction in flowering plants, this chapter begins with the structure of flower. You have studied the structure of flower in so many chapters. In morphology chapter or in plant kingdom, we have discussed it so many times, but there is no harm in revising, right? So let's start. This is the structure of a typical flower. You know very well that a flower possesses a stalk that's called pedicel, and it ends up into a swollen tip that is known as thalamus or receptacle. Right? Now, this thalamus or receptacle is actually the specialized shoot which bears various nodes in the form of a hole. So that's why from this nodes arises the leaves, okay, the modified leaves which are going to act as the parts of the flower. So these parts of the flower are going to remain in the form of a hole, one inside another, alright, and they are going to have specific functions, right? So the outermost hole present in the flower is the sepal. As a whole we call it calyx. These are mostly green in color and they are meant for protecting the flower in the bud condition. Next is the most beautiful and attractive part of the flower, that is petal. As a whole, we call it corolla. Petals, they are going to be meant for attracting the pollinating agents. That's why they are mostly very colorful, aromatic and attractive. Now, internal to the petal will remain the stamen. Also, as a whole, we call it androecium. And exactly at the center, protected by all, remains the carpel or pistil, which is also as a whole is called gynoecium. Alright. Now, sepal and petal, they are called non-essential part of the flower because they are not directly involved with gamete formation. They will help in reproduction but they will not produce any gametes as such. Hence they are non-essential. Means reproduction can happen even without them. But regarding stamen and carpel, they are the major sex organs present in the flower. The stamen is the male reproductive unit of the flower which will produce the male gametes, alright. Likewise, carpel is the female reproductive unit present in the flower which will produce the female gamete. So, because they are going to be directly involved with gametogenesis or gamete formation, we will call these parts to be essential. They are essential parts of the flower. Alright. So this is the basic structure of a flower, and it being a reproductive shoot, everything or everything related to the sexual reproduction will be involving the flower. Okay. So now, without any further delay, let us start with the male reproductive unit of the flower, that is stamen, and we'll discuss the structure and development of the stamen in details. Alright? Stamen is the male reproductive unit of the flower. It consists of two parts, filament and anther. Alright? In most of the plants or flowering plants, there are two lobes present in the anther. So these are the two lobes. Alright? And each lobe consists of two chambers. So these chambers will be at four corners and hence giving rise to tetrasporangiate state. Alright? So these chambers will be termed as pollen sac. Alright? And inside the pollen sac, there will be tiny microscopic particles that would be formed, and these are called pollen grains. Pollen grains will act as the carrier to the male gametes during reproduction. Alright? So do remember, stamen will be bilobate in most of the flowering plants, and see what basically would happen is that once the anther is fully developed, the partition wall between the two pollen sac would dissolve. This partition wall, the sterile partition between the two chambers will dissolve, giving rise to a single chamber in each lobe. This single chamber would be termed as theca. Alright? And because each lobe is possessing one theca, we will call it bithecous or dithecous condition. Alright? We will call it bithecous or dithecous condition. So now let us start with development of anther. At first, when the anther will start developing, it will consist of a circular mass of meristems surrounded by epidermis. So these are meristems and this surrounding layer is epidermis. Alright? Now these meristems, they will start to differentiate and form archisporial cell. The epidermis will remain as it is, and there will be archisporial cell that will develop. Archisporial cells. Now, the archisporial cells will undergo periclinal division. Now, what was periclinal division? Try to recall in anatomy chapter in class 11, cambial ring, it will start periclinal division. Means, it will deposit secondary xylem towards inside of towards pith, and it will deposit secondary phloem towards outside. So when two distinct layers are formed towards inside and outside, that's called periclinal division. Alright? So see, due to periclinal division, this is the epidermis as usual. Outside, a distinct layer called primary parietal cell will be formed, and towards inside, primary sporogenous cells will be formed. Alright. Now, the primary parietal cells will undergo periclinal and anticlinal division, forming certain distinct protective layers, and primary sporogenous cells will later on give rise to microspore mother cell. Now, pollen grains, another like specific term is microspore. So it is the primary sporogenous cells that will give rise to pollen grains. And primary parietal cells, they will give rise to certain layers which will protect this internal important pollen mother cells. Alright. Now, see, this circular structure will not remain circular throughout its development. There will be certain grooves, okay, that means inward constriction that will start forming in this circular structure. This inward constrictions will be very deep in the front, and on the sides it will be shallow. So in that case, finally the structure will look like this. Like this. Alright. Say this is the epidermis, outermost layer, that is going to remain as usual throughout the process. Alright, epidermis. Now, these are the primary sporogenous cells which have been scattered at four corners of this tetrasporangiate structure. And outside them, outside the sporogenous cells will remain the parietal cells, which will give rise to distinct protective layers. So what are those distinct protective layers? Just internal to the epidermis remains a single layer and this layer is called endothecium. Okay? Let me mark them. First is epidermis. Then the next is endothecium. Now, internal to the endothecium will remain 2 to 3 further layers. 2 to 3 layers of middle layers. Okay, endothecium is single layer, middle layer will be 2 to 3 layers. Finally, a very important layer will be formed by the primary parietal cells, which will remain closest towards the sporogenous cells. And this particular layer is called tapetum. Okay, it will remain closest to this primary sporogenous cells. Alright. So now let's see what are the distinct functions or nature of all these layers. Okay. First, epidermis, it is the thin and protective layer which will remain throughout the process of anther development. It will shrivel, it will dry up and it will shrivel at the time of maturity of the anther. Now, endothecium. Endothecium is a layer which is also called fibrous layer. Why? Because the cell wall of the cells of endothecium will possess some fibrous thickenings in their cell wall, and these fibrous thickenings are of alpha cellulose. Okay? So this layer, endothecium, is going to be made up of dead cells which has highly rigid and thick cell walls called fibrous. Okay, it's going to be made up of alpha cellulose. So we call it fibrous layer. Its major function is, of course, protection. Now, here there is something very important for you to understand. See, fibrous thickening will be there entirely throughout the endothecium except for one location, and that location is nearby to this shallow grooves. In this shallow groove area, here, the cells of the endothecium would not bear any fibrous thickening, so that when the anther will mature and it will dry up and it will start to break down, at that time these are the regions of the endothecium that will break or rupture. As they rupture, a pore will be formed there that will be known as stomium, and through the stomium the pollen grains, which has now fully matured, they will be released from the anther. Understood? Now, next is middle layers. Middle layers are mostly going to be nutritive in their function. They will be providing nourishment to the developing sporogenous cells. These sporogenous cells are of utmost importance because they will later on behave as pollen mother cell, and these pollen mother cells will undergo meiosis, giving rise to pollen grains, isn't it? So that's why the middle layers are the one which will provide nutrition to the developing sporogenous cells. Now, internal to the middle layers remains the tapetum. Now, regarding tapetum, there is a lot of things you must know. First, tapetum is it's a very important layer that has been formed by the primary parietal cells. It can be of two types. One is amoeboid tapetum and next is secretory tapetum. Amoeboid tapetum, these cells, they shed off their cell wall, that means they will release their protoplasm, and all this protoplasm will join with one another forming plasmodium. Okay, and this plasmodium will move towards the sporogenous cells and will be feeded upon by them. That means they will provide nourishment to the primary sporogenous cells. On the other hand, secretory tapetum, as the name itself suggests, they are going to be secreting certain important substances. So what are the secretions? Ubish bodies. Ubish bodies are certain lipid granules that contains a very important chemical that's called sporopollenin. Sporopollenin forms a part of the outermost cell wall layer of the pollen grains. The cell wall of pollen grain is mostly two-layered, outer and inner. The outer layer, called exine, is made up of sporopollenin. Now, one of the most important characteristics of sporopollenin is that it is highly resistant. There is no known enzyme yet that has been found which can hydrolyze or which can break down sporopollenin. Alright, so sporopollenin is highly resistant to temperature, acid as well as alkali. Apart from that, it also gives rise to compatibility factor proteins. See, of course, if a pollen grain of an apple tree falls onto the stigma of a mango tree, do you think fertilization will occur? No, of course not, isn't it? So, how does the pollen and the pistil know that whether they belong to each other, they are compatible to each other or not? So, this particularly checking the compatibility between pollen and pistil requires certain protein. So, these proteins to the pollen grains are provided by the tapetal cells. Okay. And finally, pollen kit. Pollen kit is a yellowish color sticky fluid which is mostly borne by entomophilous flowers, means flowers which are pollinated by insects. So this sticky substance will be surrounding the surface of the pollen grains, making them sticky so that the pollen grains can easily stick to the body of the insects which has entered the flower as a pollinator and can be dispersed to much longer distances. Alright. So this is amoeboid tapetum, this being the secretory tapetum. Now, another important point regarding tapetal cells which you must know is that tapetal cells always contain more the quantity of DNA. Either they are multinucleated or else they are polyploids. Okay, they will have higher the quantity of DNA. Now, why would tapetal cells always exhibit this character of having more DNA? That can be understood by the help of these three methods. Let's see how and why tapetal cells have more the content of DNA. Tapetal cells mostly undergo three processes. Endomitosis, restitution nucleus, and polyteny. These three processes makes the tapetal cells have more of DNA. Now, first starting with endomitosis. Endomitosis, to understand all these three, you have to be having a clear idea about the cell division chapter again. See, if you remember cell division chapter, what would happen to a cell which is undergoing mitosis? Once it successfully completes telophase, in the telophase, how will the cell look? Cell will look like this. It has two distinct nucleus formed at the two opposite poles of the cell. The only thing left now is cytokinesis, which will happen so that two distinct daughter cells are formed, isn't it? Now, if I say that a cell completes telophase successfully but does not undergo cytokinesis, what will happen? The cell will then the cell will then be in binuclear condition, isn't it? Again, the same thing repeats. It will have now 4 nucleus, no cytoplasm, isn't it? Like this, the cell will become multinucleate, and this arresting of the cell division after telophase is going to be called as endomitosis. Here, the cell will not undergo cytokinesis, hence it will become binucleate or multinucleate. Alright. Next is restitution nucleus. About restitution nucleus, again try to recall mitosis and say this time I am telling you that the cell has successfully completed anaphase and it is about to enter telophase. At that time, how will the cell look? The cell has, suppose this is one centrosome, this is another, and here the chromatids have separated, isn't it? Now in telophase, distinct nuclear membranes are to form, and in the opposite poles of the cell, two different and distinct nucleus has to be born, isn't it? Now if I tell you, instead of two different nuclear membrane, a single nuclear membrane surrounds the separated DNA or the separated chromatids. In that case, a large nucleus will be formed in the cell, isn't it? So this is going to be called as a restitution nucleus. Alright. So here in endomitosis, the cell successfully completes telophase but cannot enter cytokinesis. Here the cell successfully completes anaphase, but during telophase, instead of two distinct nuclear membrane, one single nuclear membrane will be formed. That's why there will be a resulting restitution nucleus. Okay? Next, polyteny. In polyteny, the cell undergoing mitosis will successfully complete interphase. That means S phase is complete, so the DNA has doubled up. Now the cell gets arrested in the interphase itself, cannot enter M phase. So what will happen? The cell will end up having double the content of DNA, isn't it? So that in that case we'll call the process to be polyteny. Alright? So this is how in today's class we have discussed the structure of flower. We have discussed how the anther is going to develop, and then we have discussed about what are the distinct layers and functions of each and every layer present in the anther. Finally, at each corner of the anther remains the primary sporogenous cells, which will be later on converted into pollen mother cells. Okay, and then pollen grain formation will begin. This pollen grain formation is called microsporogenesis, which will start in the next class. Alright.